These are notes based on Chapter 13 of Campbell Biology, and the topic is meiosis and sexual life cycles. And we'll talk about different kinds of sexual reproduction in different kinds of organisms, as well as the process of meiosis, which produces gametes, which are involved in sexual reproduction. Of course, living organisms definitely do reproduce after their own kind, and we're going to, as we lead into our study of genetics and heredity, then we need to understand where those gametes come from that allow for the exchange of traits in the offspring. Here we have a family. The mother in the center of the family is Sissy Spacek, the actress, who's from Texas, by the way, and this is her family, her husband and her two daughters. And you can see that even though the daughters did definitely inherit some of their characteristics from their parents, there's a combination of characteristics that are shared among them, that there are similarities, as you can see in the two daughters, with each other and with their parents, but neither one is a clone or an identical copy of the parent, and they share traits not only from one parent but from both parents at the same time. And so what we're looking at here is how those traits are passed on and how there's a mixing of those traits that are passed on in the next generation. So you acquire genes, of course, from your parents by inheriting chromosomes. That it's the genes for the traits that you inherit, not the traits themselves, even though we talk about them as being inherited traits. Genes basically are units of heredity. They're segments of DNA that code for particular proteins. Genes have specific locations on those chromosomes. Those are called loci. One singular one is a locus. And of course, DNA in most organisms is packaged into chromosomes. There are a few exceptions, but generally speaking, we're talking about chromosomes here. So the genes are going to be passed on in the gametes, the sperm and egg cells, from the parent generation to the offspring. Now, in asexual reproduction, you basically have one organism producing its own offspring by means of a process of cloning, and that can be like in plants, for instance, from a cutting. In some organisms like yeast and some other small organisms, you see budding that can occur. That's just basically a clone by mitosis, and it produces a new organism. But in sexual reproduction, you have two parents that are involved, and the chromosomes, or the genes that are inherited by the offspring, are a combination of the genes of the two parents, and so you get unique combinations of genes that are inherited, leading to a great deal of variation within the population. Here we see a hydra, a very small animal, a member of phylum Cnidaria, that reproduces asexually by budding. And also we see redwood trees, very small organism to a very, very large one. Both can reproduce asexually. Redwoods can also reproduce sexually by means of producing seeds. So lots of variation within the organisms that exist on Earth. A life cycle, of course, is the sequence of stages in the reproductive history of an organism, and sexual life cycles are based around fertilization and meiosis. In humans, we have 23 pairs of chromosomes in our somatic cells, or our body cells, and that's 23 homologous pairs. And of course, the two chromosomes in each pair are called homologous because the genes for the traits are in the same locations on this two copies of the chromosome, and one copy of each chromosome came from each parent. Here we have a karyotype, which is a picture of the chromosomes of an individual. And you can see this is taken during metaphase, so you've got the pairs lined up together. You can see the centromeres there, and you can tell that they're duplicated chromosomes. Two copies, they haven't separated yet. And so we can see that we have the pairs lined up here, and all 23 pairs are there, the 22 somatic or autosomal chromosomes, and then you've got your pair of sex chromosomes. So this karyotype is for a male because there's an X chromosome and a Y chromosome. The sex chromosomes in humans and in mammals are called X and Y. Human females have the homologous pair of those called XX, and males have one X and one Y chromosome, and then the other 22 pairs of chromosomes, of course, are called the autosomes. Each of those pairs has one chromosome from each parent, and so basically you have two sets of 23, one set that came from the mother and one set that came from the father. We call the cell with the full set of chromosomes diploid, or 2N, because it has 2 sets of chromosomes. And the gamete, or sex cell, is haploid, or 1N, and has only 1 set of chromosomes. So in humans, of course, the diploid number is 46. It's different in different organisms, and the number is really not significant. There are other organisms that have 46 chromosomes. It's not a big number, not a small number. It's just the number that we have. When you have DNA synthesis that has occurred in the cell cycle, in the S phase of the cell cycle, the chromosomes, of course, get replicated, and so the replicated chromosomes are made up of 2 identical sister chromatids. We're called chromatids as long as they're attached together, and when they separate, they become separate chromosomes. So here we have a cell with 1, 2, 3 pairs of chromosomes here. So the 2N number is 6. Okay, there's 3 chromosomes from the mother, the red ones, and 3 chromosomes from the father, the blue ones. And you can see here that these chromosomes have been duplicated. They're each made of 2 sister chromatids, and you can see these pairs that are there, these homologous pairs that are present in the cell. All right, a gamete is going to have only 1 set of chromosomes, and so we have to have a way to get to that 1 set of chromosomes, and that's the process of meiosis. In egg cells, the sex chromosome is always going to be X, since the mother has 2 X chromosomes, and that's the only kind of sex chromosome that she can contribute. In a sperm cell, the sex chromosome can be either an X or a Y because the father has both. Now, in the human life cycle, the union of gametes, of course, is called fertilization. Fertilized egg is called a zygote, and it has 1 set of chromosomes for each parent. It's a diploid cell, and then it produces somatic cells by mitosis as it develops into a child and then into an adult. At maturity, at sexual maturity, then the gametes can be produced by the sex organs, or the gonads of the person, or the animal, or the organism. In humans, the gametes are the only human cells that are produced by meiosis. All other cells in the human body are produced by mitosis. In meiosis, we're going to get 1 set of chromosomes in each gamete, and then we have the alternating fertilization in meiosis in the life cycle to maintain that chromosome number. I often tell the younger students that you wouldn't want your offspring to have 92 chromosomes, but so you want the offspring to have the same number of chromosomes, so you have to cut the number down if you're going to combine 2 cells together. Here we have a representation of the life cycle of human, alternating between meiosis and fertilization here. And so most of the life cycle you can see is diploid, and the only haploid portion of the life cycle is the gametes, the haploid gametes, the egg cell and the sperm cell. When they unite by fertilization, they produce a diploid zygote, which undergoes mitosis and development to develop into the adult, which can then later during the fertile years produce the sperm cells and egg cells that can be used for reproduction. Now, it's different in different kinds of organisms because there are three different types of sexual life cycles, and that's based on how the meiosis and fertilization are timed compared to each other. So we'll talk about those other life cycles briefly in this unit, and then we'll talk about them again when we talk about those particular groups of organisms. Animals, of course, the only haploid cells are gametes, okay, and they're produced by meiosis and then undergo no more cell division before fertilization occurs, and then they produce the zygote that produces the multicellular organism, like we talked about with humans. This is the generalized life cycle that you'll see for animals. Okay, you see that you have 2 different kinds of haploid gametes that can combine together in fertilization to make that zygote, which grows into the diploid multicellular organism. Plants and some algae have this alternation of generations that includes both diploid and haploid cell stages that are multicellular. The diploid organism in plants and algae is called the sporophyte, and it makes haploid spores by meiosis. And then the haploid organism is called the gametophyte. Okay, the spores grow into a haploid organism. That's the gametophyte, and then it makes haploid gametes by mitosis, I'm sorry, and then fertilization of those gametes results in a diploid sporophyte. So here we have This is a moss, and so it shows you basically what we have in a moss because it exhibits real easily the two different parts of the life cycle. So the diploid phase of the life cycle is called the sporophyte, and the sporophyte produces spores by meiosis. The spores then can grow into the multicellular gametophyte organism, which produces the gametes by mitosis. Since it's already haploid, it doesn't have to undergo that reduction division to produce its gametes. But when the gametes combine together to make the diploid zygote, then that's what you get when you get the sporophyte. In plants that you're familiar with, like trees and bushes and flowers, the plant that you recognize is the sporophyte part of the plant, part of the life cycle, and the gametophyte in seed plants and flowering plants is much reduced to only be part of the production of the seeds, basically. Now, it's different in fungi. In most fungi and in some protists, they have only a very small diploid stage. That's the single-celled zygote. There's not a multicellular diploid stage. Then the zygote produces haploid cells by meiosis then, and then those grow into a haploid multicellular organism, which produce gametes by mitosis. So this is two different kinds of life cycles where the gametes are produced by mitosis rather than meiosis. Here we have the diploid zygote, which undergoes meiosis to produce the haploid, or the spores, I guess, that grow into the haploid unicellular organism or multicellular organism in the case of fungi. And then it undergoes mitosis to produce gametes when it's time to combine gametes and exchange some genes. So a little bit different kind of thing. And again, we'll talk more about those when we talk about plants and about fungi later on. Either of those types of cells can divide by mitosis, but only diploid cells can undergo meiosis. And so making sure that you have that, the halving or cutting in half of the chromosomes, chromosome number, or doubling the chromosome number, allows for a great deal more genetic variation in the offspring, which of course is beneficial to the species as a whole. Meiosis is the way that we can reduce that number of sets from diploid to haploid. And this is a type of cell division similar to mitosis in some ways, but very different in other ways. Like mitosis, there is a replication of chromosomes. There is a normal cell cycle that precedes the division phase of the cell cycle. And it includes an S phase, which is going to be the replication of the chromosomes. And then we've got in the division part, in the M phase of the meiosis cell cycle, we have two sets of divisions called Meiosis I and Meiosis II. They're going to produce at the end 4 daughter cells rather than the 2 daughter cells that you get from mitosis. And each of those daughter cells that eventually result will have half the chromosome number. So we've got two divisions that follow the doubling of the chromosomes. Okay, Meiosis I is the reductional division where the homologous chromosomes pair up and separate, and that produces 2 haploid daughter cells, but the chromosomes they have in them are duplicated or replicated chromosomes. And then the second division is called equational division, and in that one the sister chromatids separate to produce eventually the 4 haploid daughter cells. Here we have just in pictorial form to see that you've got your homologous chromosomes in the parent cell. It's diploid. We've gone through the cell cycle, through the S phase of the cell cycle, and duplicated those chromosomes. And so here are our chromosomes that are duplicated. Then in the first division, those homologs separate from each other. So we have each of these haploid cells has duplicated chromosomes. And in the second division, then those chromatids separate, and that gives us 4 separate daughter cells, each with half the chromosome number. Meiosis I, like we said before, is definitely preceded by an interphase, and that's when the chromosomes get duplicated. And then, generally speaking, they're identical to each other, joined at the centromere, and we have the introduction of the cell division process. So 4 phases in the first division of meiosis: prophase I, metaphase I, anaphase I, and telophase I, followed by cytokinesis. Okay, and this gives you an overview of basically the whole process. In Meiosis I, we're going to separate the homologous chromosomes. In Meiosis II, we're going to separate the chromatids. So in prophase I, similar to prophase and mitosis, you have the disappearance of the nuclear membrane, the production of the spindle there, but something that's unique in meiosis is you have the pairing up of the homologous chromosomes, and when they do, they're able to cross over there in these little X shapes there and exchange parts of their chromatids with each other. And we'll talk about that in just a minute. Okay, in metaphase 1, those homologous chromosomes pair up line up along the metaphase plate. Each one of the centromeres, the centromere for the blue chromosome and the centromere for the red chromosome, are going to attach to only one spindle fiber at the kinetochore rather than two. And so when anaphase occurs, that's going to pull one chromosome to one direction at one end of the cell and the other chromosome to the other end of the cell. So we end up with these two haploid cells that each have duplicated chromosomes, but half the chromosome number in general. There is definitely a cytokinesis that occurs here. There's also, as you can see in the picture here, a decondensation of the chromosomes back toward the chromatin and development of a new nuclear membrane at the same time as the cytokinesis is occurring. Prophase I is the longest phase of this whole process. It typically takes about 90% or even more of the time required for the entire meiotic process. So in prophase I, you've got the condensation of the chromosomes, then the homologous chromosomes pair up in a process called synapsis. They're aligning themselves gene by gene. And then when they cross over, the non-sister chromatids can exchange segments of their DNA. They form this structure called a tetrad. Tetra means four, and so you've got a group of 4 chromatids that are there together. And usually they have at least one, if not more, chiasmata. One is called a chiasma. Multiple ones are called chiasmata, and those are X-shaped regions where the crossing over occurs. In metaphase 1, the tetrads line up at the metaphase plate, and there you have microtubules from one pole attaching to one chromosome and from the other pole attaching to the other chromosome at the kinetochore. And then in anaphase 1, those pairs of homologous chromosomes are going to separate, moving one chromosome toward each pole, and the sister chromatids remain attached to each other at the centromere and do not separate in this part of the division. Telophase 1 in cytokinesis, we have a haploid set of chromosomes in each one. The cytokinesis happens usually simultaneously with telophase, and that forms these 2 haploid daughter cells. And again, they do start forming nuclear membranes, and they do start decondensing into chromatin before proceeding to the second division. In animal cells, you've got a cleavage furrow. Of course, in plant cells, you've got a cell plate like we've learned about before. But here's where the big difference is. Another big difference with mitosis is that we don't have another replication that occurs before the next division. In Meiosis II, we also have 4 phases: prophase II, metaphase II, anaphase II, telophase II. And this part of the meiotic cycle is very similar to what you see in mitosis. So here we have again, the reabsorption of the nuclear envelope and the condensation of the chromosomes, then the formation of the spindle in prophase I, like you are accustomed to seeing in mitosis. In metaphase I, those chromosomes line up on the metaphase plate, and this time the kinetochores of each chromatid attach to one to each pole. And then in anaphase II, those molecules that are holding the centromeres together separate and allowing the sister chromatids to separate into separate chromosomes. And finally, in telophase II and cytokinesis, we end up with 4 total separate daughter cells that are haploid in number, each having single chromosomes. The spindle apparatus forms like we saw before, and then the chromosomes move toward the metaphase plate as they're attaching to the spindle fibers. They're in metaphase II, of course, they're arranged at that plate in the middle. And here's where you see a big difference, because since they crossed over in prophase I, the chromatids of each chromosome are no longer genetically identical. They have exchanged portions of their chromatids with each other, and so rather than being all of mom's chromosome or all of dad's chromosome, they have some of both on each chromosome. And then the kinetochores attach to microtubules, and then they separate and move separately as individual chromosomes to the poles, where the new nuclei form and the chromosomes begin decondensing into chromatin. So each daughter cell here at the end is genetically distinct from every other one and from the parent cell. Now, mitosis, remember, conserves the number of chromosomes. The products of mitosis are exactly like the parent cell, and they have the same number of chromosomes. They're identical to each other and to the parent cell. In meiosis, we're reducing the chromosome number from diploid to haploid, and that's going to produce haploid daughter cells that are different from each other and from the parent cell. Here we have a summary of the processes comparing mitosis to meiosis here, and you can see the differences that are occurring. They both can start with the same kind of parent cell, but you're going to have dramatically different results at the end. This chart also is a nice comparison showing you kind of what occurs in terms of these different processes, DNA replication and division, of course, of what happens in the production of the gametes and of the somatic cells and the roles that they play in the animal body.